Well, aloha everyone, and welcome to our lecture on the digestive system. This video will talk about the different anatomical components of the digestive system and their respective functions, how they interact with one another, how they're regulated, mainly neuronal and hormonal control. We will talk about the difference between mechanical and chemical digestion. And last but not least, we'll talk about what happens as we get older or end up with a disease state in the digestive system. So this is an overview of the organs of the digestive system, and anything that you see that's shown in red is going to be considered an accessory to the digestive system. So not an actual digestive system organ, but an accessory organ. And basically what happens is food's going to enter into the mouth and oral cavity where it's going to get what we call masticated or chewed up. We're also going to add saliva and salivary enzymes here. And then it's going to get swallowed, so the bolus of food goes down the esophagus, where it's going to enter into the stomach. The stomach here is depicted in dashed lines because it's actually hidden here by the liver. So it's going to enter into the stomach. As it passes through the stomach, it's going to enter into the small intestines. As it enters in the small intestines, again, that's all kind of depicted in lines here because it's behind what we're seeing here, the transverse colon and the liver. So it's going to end up with a bunch of enzymes that are added in from the liver and that are stored in the gallbladder and released into the duodenum of the small intestines. Then we're going to go through the small intestines where the digestion is going to continue, and then eventually we're going to end up into the large intestine. So here we have the ascending, the transverse, the descending colon, and then the spent food will be released through the rectum and out through the anus. And this is what this looks like in an organism. So here we have the liver. Remember, the liver is held in place here by here's a falciform ligament. Behind the liver is the stomach. Right? Food that passes through the stomach is then going to enter into the small intestines. Again, the small intestines is kind of hidden for a short while. Then we enter into here the duodenum of the small intestines, which we can visualize, then through the ileum. Then we're going to head up into the large intestines. You can see here the ascending colon, transverse colon, descending colon, and then again out through the rectum. So it's kind of an overview. Don't worry, we're going to talk about each and every one of those organs on its own little slide as we pass through here. So the gastrointestinal tract is basically one long tube that's going to be open at both ends, right? Food goes in, waste goes out, and during the transit through this tube, the food is going to be processed. Some major portions of these tubes that you might recognize include the esophagus, right? Your throat, where you swallow, your stomach, where the food enters into, and then the small intestines, where the food passes after it exits the stomach. Then it passes into the large intestines, and then finally out through the rectum. Now we have several accessory structures that we're going to talk about throughout this lecture. And accessory structures are not part of the GI tract themselves, but they do greatly enhance and contribute to food processing. So that's going to include things like the teeth for chewing, right? The tongue for moving the food around and swallowing the bolus of food. Salivary glands, which is where we're going to first start chemical digestion. We're going to start breaking down some sugars in the saliva. So we have some enzymes in there called amylase, which breaks down amylose, which is in the sugar, the carbohydrates. We also have the liver, which is going to create bile, which is going to get stored in the gallbladder, and that's going to get secreted into the duodenum of the small intestines. We also have the pancreas. At that same intersection, we're going to be secreting some pancreatic enzymes, which are going to help with digestion in the small intestines as well. So the overview of digestion includes 6 basic processes. The first is ingestion, or just the process of putting food into your mouth, and then you're going to start chewing that food up. And that's going to cause the secretion of different enzymes into the lumen of the GI tract. And that's going to start all the way in the mouth and continue through into the stomach and into the small intestines, etc. The third thing that has to happen is all of that food and enzymatic material has to be churned. So we have to have mixing and propulsion. So it's going to be churned as it moves through the GI tract. And this is going to lead to digestion, which is number 4. Digestion is 2 different sections, both mechanical and chemical breakdown of food. So mechanical will be something like chewing or mastication or physical breakdown. And chemical breakdown involves enzymes or acids or things that are going to help chemically digest the food themselves. We also have to have the absorption. The entire point of digestion is to be able to get the nutrients that our body needs, and that's going to happen mainly in our small intestines. So digested products from the GI tract are going to get picked up by the blood and the lymph that surround the small intestines and then get back into the general circulation to be able to feed and nourish the rest of the body. And then last but not least, whatever is not absorbed is going to have to be compacted. We're going to be removing a lot of water at this point as well and making it hardened into a fecal product, which is going to end up defecated out from the GI tract. Alright, so this is an overview of the different layers of the GI tract, and you're going to see similar but modified images as we travel throughout the GI tract. So I'm going to start in the center here. So in the center here we have the lumen. So this is where your food is going to be, right? Surrounding that we have something called the mucosal tissue. And we have 3 separate layers to that. This is the epithelial layer here. Surrounding that we have the lamina propria shown in light pink. Surrounding that we have the muscularis mucosa. Before I move on from this section, I want to point out a couple of important things. One here, these here are these lymphatic tissues. These are MALT or mucosa associated lymphatic tissues. And this is going to help take on any bacterial invaders that may be coming in through the food supply. We also have glands here, both in the mucosa and in the submucosa, that are going to be secreting different enzymes, etc., into this food as it travels through the digestive tract. Okay, going outward, we have the submucosa, which sits right around inside the mucosa. Again, those are going to have glands inside here as well that are going to be secreting into the lumen. That's going to be innervated, right? That's innervated here by the submucosal plexus. It also has veins and arteries that are going to feed it, so it's going to have blood supply. Surrounding that, we have the muscularis. The muscularis here has 2 different layers, a circular muscle and a longitudinal muscle, and when we enter into the stomach, we're going to add into a 3rd layer. And what happens basically is we have these muscle layers that are going to be able to contract in different directions. And this is going to allow us to both squeeze the food forward and also to churn it in place. Inside this muscularis layer, we have another plexus. This is the myenteric plexus. This is going to have different set of innervations. They're going to be linked so they can work in concert, but they're also separate so that they're able to work independently of each other. Surrounding that, we have the serosa layer and then the epithelial layer. Okay, so the enteric nervous system is regulated by 2 different plexus, as I just mentioned, the submucosal plexus and the myenteric plexus. And it's also regulated by the autonomic nervous system. The autonomic nervous system, as you may remember, is separated into the parasympathetic and the sympathetic divisions. And as you may understand, fight or flight is an opposite regulation than rest and digest. So the GI pathway is going to be regulated by a bunch of different reflex pathways that are going to regulate the secretion of different enzymes into the lumen, and also the motility of the GI tract is also going to respond to different stimuli that are going to be given to this system. Let me step back for a second and start talking about some of the digestive organs. So here we've taken a mid-sagittal plane and we're looking inward into the center of the abdomen. If we're starting at the top here, something you may recognize is the diaphragm. So above this is going to be where we have our lungs. It's going to be assisting us with breathing. Underneath that, we have the liver. Remember, the liver is held by the falciform ligament, which is not shown here. This here is the lesser omentum, and you're going to see several sets of the omentum. So we have the lesser omentum and the greater omentum, and these are going to be connective tissue. On top of that, we have the parietal peritoneum. So the peritoneum is another set of connective tissue. So these are going to be connective layers that hold all of these organs in their respective locations. All right, this here is the stomach. Hold on. This here is the stomach. From the stomach, we're going to be passing into the duodenum of the small intestines, which is depicted here. And then that's going to then go through the rest of the small intestines and then end up entering into the large intestines. We're going to enter into the ascending colon, which is not depicted here. This here is the transverse colon, which is going to be cut. And then it's going to head down on the opposite side into the descending colon and then out through the rectum. All right, so here's the liver with the falciform ligament. In most of our depictions from here on out, the liver is either going to be folded upward or removed entirely from our depiction so that you're able to see what happens behind it. Behind that, we have the stomach. And the stomach is, remember, one of the first places that the food is going to enter into after we've chewed it and then swallowed it. After the stomach, the food is then going to enter into the duodenum of the small intestines. It's going to enter all the way through the rest of the small intestines, ascending, so now we're into the large intestines, ascending, transverse, descending colon. And you're going to hear me say these in order over and over again. Another thing I want to point out here, this little green organ is the gallbladder. And it's visualized here as a little nubbin when the liver is down, but when the liver is lifted upward, you can see it. And this is an organ that's going to store the bile that the liver produces. So it's another accessory organ of the GI tract that's going to secrete the bile that the liver produces when necessary into the duodenum of the small intestines. Remember we talked about that omentum? The omentum here is this connective tissue on top, and again, it's going to be an area where all of the different organs are going to be kind of anchored down. Oftentimes we're going to have that removed so that we can see what's underneath. So here's our greater omentum. It's going to be pulled upward. It's going to be obscuring our view of the stomach and the liver behind it. Inside here, we're looking at the small intestines. Here's the duodenum that's been pulled laterally so that we can kind of see behind that. This here is the mesentery. It's another set of connective tissue. It's where everything's going to be kind of pinned down to. Here we have our ascending. Again, it's going to be behind the duodenum, which has been pulled over. Ascending, transverse, and descending colon. Then here, out through the sigmoid colon, and then out through the rectum. This is what it looks like in an actual organism, so you may recognize these here are the lungs and the heart. Here's the diaphragm. We're actually going to be looking a little bit lower here for the digestive tract. Again, here's the liver. The liver's got this falciform ligament that connects it. Behind that, we have the stomach. You can't really see much of it, and then all of this is the omentum, which is that connective tissue. Again, you won't be able to see anything. It's going to obscure our view. All right, so the first thing that the food is going to enter into the GI tract is going to be the mouth. And the mouth is going to be formed by major structures, including the cheek, the hard and soft palates, the tongue, and then of course the teeth are going to be found inside here as well. Some areas of interest, this is the frenulum. We have several of those. It just means connective tissue. This is what gets cut when an infant has something like a tongue, this would be the tongue tie or a lip tie. Here's the labial frenulum, which is the top, or the inferior labial frenulum at the bottom, the lingual frenulum underneath the tongue that allows for the tongue to move around. If we move inward inside the mouth, we have the hard palate here. It's quite bony and it's going to be covered in gum. Right? The soft palate here is not bony, it's muscular, it forms the rest of the roof of the mouth, and then it goes down here into this little area here, which you may call the dangly thingy at the back of your throat. That's called the uvula, and that's going to help prevent food that's going to be swallowed from entering into the nasal cavity, so it doesn't flip up here above the soft palate and end up in the sinuses. Continuing backward here, we have these guys here. These are the fauces. We have two different arches. This is the palatoglossal arch here. And then there's another arch here in the back. That's the palatopharyngeal arch. And around that we have the palatine tonsil. And you may remember when we talked about the lymphatic system, how the tonsil tissue is going to be surrounding the area of the throat and is going to be one of the first areas of immune defense for anything that's ingested. So this is going to be part of our tonsil ring here, this palatine tonsil. Then these guys are gums. You may see them gingivae. So if you've heard the word gingivitis, that means gum infection. Itis obviously meaning infection. What else am I missing here? Oh, and the different types of teeth: molars, premolars, canines, and incisors. All right, one thing we haven't talked about here are salivary glands. Salivary glands lie just outside the mouth, and they empty their contents, so saliva, through these ducts that are going to enter into the oral cavity. And we have 3 pairs of them, so 6 total. Right, 3 pairs: parotid, submandibular, and sublingual. They look like this. Here's our parotid up here, submandibular, and sublingual, so under the tongue, around the mandible, and then this one's going to be up here, kind of by the ear, but it's going to feed into this parotid duct, which opens back in the molars in the mouth. And if you look at these underneath a microscope, you'll see that they have these acini regions that are going to create serous or blood supply, and then mucus. And mucus and blood supply are going to be really important here inside the mouth, because this is going to be the first areas where we're actually digesting. So we're secreting digestion enzymes, and we're also going to be picking up some things that are going to be entering into this blood supply. So it's going to be a connection, but the very first connection between the food and the body. All right. The tongue is a muscle that you're obviously very familiar with. I'm using it right now to speak to you. It's going to be associated with sets of muscles that are going to help form the floor of your oral cavity. It's basically one large muscle that's covered in a mucous membrane and helps with things like chewing, swallowing, speech, etc. And on top of it, it's covered with papillae. And we talked about papillae and taste buds when we talked about the special senses lecture. But just to familiarize you, these taste buds are what are going to interpret the signal of whatever food is in your mouth into something like sweet or salty or umami or bitter, and then send that information into the neural passages into your brain so that you can discern what we consider to be taste. Okay, so the teeth are going to be responsible for mechanical digestion, the grinding of your food during what we call mastication or chewing, and they have sections, right? So crown is that top section, the neck is that middle section, and the root is the very bottom. So if you've ever had a root canal drilled into here and probably eliminated the nerve so it doesn't hurt, and then packed it all back in and then put a false crown on top. This is why when we have dental work done, we call it putting on a crown, because you're basically replacing the crown that is already presently there. And that crown consists of enamel, which is one of the hardest parts of the body, and dentin, which is calcified connective tissue. The dentin is actually going to make up the majority of the tooth, so it's going to run all the way down into the root. So it's what's protected by the crown, but it's going to be surrounding this pulp cavity. And inside the pulp cavity, we have nerves and blood vessels and tissue. And then surrounding this whole thing, so basically coming right underneath the enamel, headed down to the root of the tooth, we have something called cementin. And cementin is this substance is very similar to bone and is actually going to be attaching the root of the tooth to the periodontal ligament, which is depicted here. The periodontal ligament obviously is going to help anchor the tooth to the underlying bone. And then at the very bottom here we have foramen. Foramen just means hole. Apical would be the apex. So the apical foramen is a hole that's going to allow blood vessels, lymphatic vessels, and nerves to enter and exit the tooth. Underneath this we have a series of nerves and a lot of blood supply. This is why the mouth tends to be a major series of bleeding if you end up injured, because you have a lot of blood supply in that area. I'm just kind of going to gloss over this, but basically what this slide is showing is that as a child you have a different set of teeth than you do as an adult. So these are called your milk teeth, your primary teeth, your deciduous teeth. You may have heard them by a bunch of different things. Basically they're your children's teeth. And then as you get older, the larger teeth, which are already inside of your jaw as a child, actually a child's skull is very creepy because the teeth are already up inside waiting to push through. But basically they're going to push through and they're going to push these teeth out and then be the replacement teeth as soon as your mouth is growing and large enough to handle it. And they come in at different times. So you may notice here that the central incisors come in at 8 to 12 months. These are going to be one of the first sets of teeth that a baby gets, right? These first 4, 1, 2, 3, 4. Then surrounding that, we're going to get the lateral incisors. So then eventually they're going to get 4 on top and 4 on bottom. And that happens around the 1 year mark. And then just a little bit after that, they get their molars. So they actually are going to skip these. They get these molars here around the 12 to 16 month mark. And then later on, they get this 24 to 32 months, around the 2 to 3 year mark, they end up with a second set of molars. And then kind of the same thing happens as a child goes into their adult teeth, so they lose them in sequence. So first we're going to lose some like the central incisors, etc. The first molars are going to come in around 6 to 7 years. So they're going to be replacing this original molar and popping them out. So some of the first teeth that a child is going to lose are going to be this first front teeth and then some of these back teeth as well. And then they're going to start coming in again in waves. So the first ones come in around 7 to 8 years of age, and then the later ones come in around 11 to 13 years of age. And then the last set, those wisdom teeth, don't generally come in until 17 to 21 years of age. And some people don't get the full set of wisdom teeth at all. Overall, as an adult, you're expected to have 32 healthy teeth. But as a child teeth, we're going to label them as A, B, C, D, all the way to T, just to be able to differentiate on the dental records. All right, so what's the responsibility of teeth? Mechanical digestion, right? They're responsible for chewing the food. That's going to help mix the food up with some of the saliva. Remember, saliva has salivary enzymes in there, specifically amylase, which is going to break down amylose being one of the sugars, and it's going to form what's called a bolus. That bolus of food is what you're actually going to swallow, so the tongue's going to force that bolus of food to the back of your throat, and then you're going to swallow it. It's going to go down the esophagus into the stomach, where it's going to continue with the digestion, specifically from that point forward, mainly chemical digestion. One of the first sets, again, of chemical digestion is going to occur in the mouth by that salivary amylase, which breaks down amylose. So it's going to break down polysaccharides or long chains of saccharides into disaccharides or 2 chains. All right, so what happens in the mouth? Just to summarize, the cheek and lips are going to help keep the food in between the teeth. The salivary glands are going to help secrete saliva. That's going to help moisten, dissolve food. Also helps keep your mouth and teeth kind of clean and has some antibacterial properties as well. Your tongue does a lot of things, speech, etc. But in terms of digestion, it's going to help with moving that bolus of food backwards. So it's going to help maneuver it around during chewing or mastication, shape it into that bolus, and then help you swallow it. We also have taste buds on there, which are going to help interpret what you do and don't like to eat. And it's all going to be converted into neural signals. So it's going to be relayed back to the brainstem, basically. And then lingual glands, which are going to secrete lipase. Lipase is going to be one of the very first digestive enzymes to start breaking down the fats. So lipase breaks down lipids. Anytime you see ACE, breaks it down, lip being lipids. So we're going to be taking triglycerides and breaking them down into fatty acids and diglycerides. And you'll notice as we pass through the digestive tract, one of the first things we do is we break the big macromolecules into smaller ones. And then later we break the smaller ones into smaller and smaller ones. So often what you'll see is a polymer broken down into a dimer. And then later we'll see the dimer broken down into a monomer. Okay, so again, the food is going to enter here into the oral cavity, then be swallowed by the tongue and go down the esophagus. So what's it going to pass through? This is the region called the pharynx. The pharynx is a large tube. It's a tunnel, basically. A funnel, basically, that's going to extend from the nares, or the nose, down through the esophagus, and also to the larynx. So this is kind of an overview of everything that's going to be in the pharynx. We have 3 major regions: the nasopharynx, the oropharynx, and the laryngopharynx. So the nasopharynx is going to be everything from the nares region down, including the pharyngeal tonsil region down. That's going to include the opening of the auditory tubes. This is actually going to help go up into your ear. So when you are equalizing pressure, for example, that's going to be going through the pharynx up into the auditory tube. At the back of the mouth, we talked about the uvula and the palatine tonsil. We also have this region here called the fauces, which is going to be at the very top of the tongue where it connects to the uvula during the act of swallowing. Here's the oral pharynx, which is just going to be the oral cavity, right, the mouth cavity of the pharynx region, and then the laryngopharynx, which is going to be where our vocal cords are, or our larynx. The vocal cords here are going to be down here. We have a true vocal cord and a false vocal cord, so a vocal fold and a vestibular fold. And then underneath that we have the larynx, and that is going to be surrounded by a lot of cartilage. So you'll know in your throat we have several sets of rings of cartilage. That's all for the trachea. So this is not involved in digestion, right? This is involved in speech and respiratory pathways. So what keeps the food from going down the trachea? I'm glad you asked. That's this little thing here called the epiglottis. So this epiglottis here is normally going to be up, which is the open position. That's for breathing. Air goes in, air goes out, air goes in, air goes out. Oh wait, food goes in the mouth. Well wait, we have to swallow. This folds over so that the bolus of food will pass past the trachea and go down the esophagus. All right. So the esophagus is basically a muscular tube. It is collapsible. Unlike the trachea, remember the trachea has a lot of cartilage in it. It lies behind the trachea, and basically it's always closed until you swallow. And then it's open for the time frame for the bolus of food to go down, and then it's closed again. And its main objective is basically just to connect the pharynx to the stomach. Now, just to show you an overview of the layers of tissue that we talked about previously, again, we're going to be working from the lumen on our way outward. Here we have the mucosal layer, which has the epithelial layer, the lamina propria, and the muscularis mucosa. Then we have the submucosal layer, then we have the muscularis layers, and the adventitia. The process of swallowing is called deglutition. So first we have mastication or chewing, then deglutition or swallowing. Basically what happens here, we have this bolus of food which is going to pass back through the soft palate, past the uvula when the tongue forces it backwards during the act of swallowing. It's going to enter into the oral pharynx. At the same time, this region here, this epiglottis, is going to fold over. You can see it was depicted previously as just one line. It's actually kind of two. It has a folding hinge here. So it's all going to fold down so that the bolus of food can go past it. At this point, the uvula and the palate are going to seal off the navel cavity so no food can go upward. So food, after being swallowed, only has one option, and that is down the esophagus. At this point, breathing is temporarily interrupted, and you can see why, right? No air is going to be going in and out of the trachea during that swallowing moment. This is what this looks like. Here's the esophagus. The esophagus has different sets of muscular layers. When they're relaxed, it's going to be open but flattened. When it's contracting, it's going to be pushing the bolus of food down until it reaches what's called the lower esophageal sphincter. And this is basically like the door between the esophagus and the stomach. And if you've ever gotten to the point on Thanksgiving dinner where you feel like you're so full that you just can't eat another bite, well, that's because you probably truly cannot, right? The sphincter is simply just not allowing any more food in, and now it's sitting at the top of your esophagus, or the very bottom of your esophagus at the top of your stomach, which is very uncomfortable. What's interesting about swallowing is that you can do it even laying upside down, because these muscles are going to contract in concert, and so it's not gravity fed. So you can do it laying down, you can do it hanging upside down. So what happens in the pharynx and esophagus? Well, basically, we're going to be moving the bolus of food from the mouth, or the oral pharynx region, down to the laryngopharynx region, which is going to pass through the trachea, so we're going to have the epiglottis moving over. That's going to close off the air passageways. In the esophagus, we're going to have something called peristalsis, which is the esophageal stage of deglutition, which is going to push that bolus of food down the esophagus until it gets to the stomach. Now, before it gets to the stomach, it has to knock on the door of the lower esophageal sphincter, and that's what's going to be allowing the food into the stomach. The esophagus also secretes mucus, which helps lubricate the food, or the passage of the bolus of food throughout the esophagus. All right, so what does the stomach look like? So here's the esophagus. It leads into the stomach. We have to knock on the door of the lower esophageal sphincter to get into it. We actually have a lot of specialized regions. So this region is called the fundus. You're going to hear the word fundus several times throughout these series of lectures. It just means the opposite opening from the opposite cavity from the opening. So we have a gallbladder fundus, for example. We'll have a uterine fundus, for example. It's just going to be whatever region is going to be the farthest away from the opening, or opposite. So here's the fundus, which is opposite the opening. We have the greater curvature here and the lesser curvature here, so you can, big spoon, little spoon. And then here we have the ruga inside. And the ruga mucosa layer is really important because, and you'll see this again in the urinary system, we talk about it in the urinary bladder, the mucosa is important because it allows for expansion. So when you add a bunch of food to it, the stomach is going to be able to get larger and larger and larger to accommodate much more space than it normally is going to take up when it is contracted. Okay, now I mentioned that we had 3 layers of muscle here, where elsewhere we only had 2. So we have an entry of a new layer here. This is called the oblique layer. We only see it in the stomach. So we have the longitudinal layer, the circular layer, and the oblique layer. And in this way, we're not only going at 90-degree angles to each other, but we have a third direction that we can pull. So you can see this muscle layer, for example, is contracting this way. This one's contracting that way, and this one's contracting this way. So they're all going to have different directionality of contraction, which allows for all of the churning. All right, as this bolus of food enters into the stomach, it's going to then become chyme, chyme, I guess. As chyme is going to be secreting from the stomach, it comes in small little blebs. So although you might eat a whole Subway sandwich all at once, it's going to come out in small little packets of food that's going to be passed into the duodenum, which is the very first portion of the small intestines. In order to do that, first it has to go through this sphincter here. This is the pyloric sphincter. So the sphincter at the top of the stomach is the lower esophageal sphincter. At the bottom of the stomach is the pyloric sphincter. This is the pylorus region. Again, this enters into the duodenum of the small intestines. And when we talk about the liver and the gallbladder and the pancreas and how they secrete these different things like bile and digestive enzymes into the duodenum, they're going to be secreting them right here. Why? Because as you may imagine, the stomach is very, very acidic. So inside the stomach, we have a very low pH. But as soon as we enter into the small intestines, we want to restore a normal pH of approximately 7. So it's really important that we add things back into the intestines that are going to help restore our intestinal pH. At the same time, we may know that certain enzymes only work at certain pHs, which means that we have an enzyme that's very specific to the stomach. But as soon as it enters into the small intestines, it no longer works. So we have to have an alternative to that in the small intestines. And again, that's going to be coming from secretions that come from things like the pancreas, etc. This is what it actually looks like inside an organism. Here's the esophagus. As you head down the esophagus, you're going to reach that lower esophageal sphincter. And this is the stomach itself. The stomach has the fundus region, the cardia region, the body region, again with the lesser and the greater curvature, and then a bunch of ruga mucosa in there that's going to allow it to expand. As we enter into the pyloric region, we enter into the pyloric antrum, and then eventually into the pyloric canal, then the pylorus itself, and the pyloric sphincter allows food to exit in small little boluses, now this is called chyme, into the duodenum, where we can continue and move on with the next set of digestion. So what's the point of the stomach? What are the purposes of the stomach? Well, the stomach, first and foremost, is going to get our pH really low. It has HCl inside the gastric juice, and this is responsible for killing bacteria and denaturing any proteins. It's also going to help begin the digestion of proteins using something called pepsin, which is that specialized enzyme that only works at that low pH. So what the point of the stomach is to mix saliva and food and gastric juice to form what we call chyme. Chyme is what's going to be secreted in little blebs into the small intestines. It also serves as a food reservoir before release into the small intestines. Again, you don't sit there and nibble on one bite all day long. Instead, you're going to sit down and eat an entire Subway sandwich, and then the stomach is going to dole out one bite size of chyme into the small intestines at a time. Another thing that the stomach does is secrete something called gastrin. Gastrin is going to go into the blood supply, and it's going to help regulate the different uptake and release of different molecules for digestion. So if we look at the histology of the stomach, we're going to see that those different layers that we talked about, the mucosal layer, submucosal layer, muscularis, and serosal layer, are going to be specialized. And we'll see that as we travel through. We'll get more different specialization in the small intestines and then different specialization in the large intestines and the colon. But in the stomach, we have something called gastric pits. So this mucosal layer makes these gastric pits, which are going to secrete things into that bolus of food, which is then going to become chyme. We have mucous neck cells here, which are going to be right at the top of these deep pits. Inside the pits, we have parietal cells, chief cells, gastric cells, and G cells, which all secrete different things, and we'll go over them independently. In that submucosal layer, we are going to have these lymphatic modules, the MALT, so the mucosal-associated lymphatic tissue. We also have lymphatic vessels. Lymphatic vessels here are shown in green, and what's interesting about lymphatic vessels here is that, yes, they are part of the immune response, absolutely, but they are also a part of the uptake of fatty acids. Fatty acids and certain things are going to be too large or too hydrophobic to interact with the blood supply, and so they're going to have to go through the lymphatic system and then get dumped in through the hepatic portal vein and eventually make it back into the circulatory system at a different region of the body. So the lymphatic tissue is actually also responsible for the uptake of different hydrophobic molecules. All right, again, underneath here we have the muscularis layer, and this time we're going to have a unique layer here. It's called the oblique layer. So we have the oblique layer, the circular layer, and the longitudinal layer. And you'll see the circular layer and the longitudinal layer in almost all of the different sections of the GI tract, but you're only going to see that oblique layer here. And that allows for contraction in not just 2 directions, but in 3 directions. So we can end up churning all of the contents of the stomach up to be able to allow all of those digestive enzymes to enter into every piece of food. So this is what this looks like kind of up close. I told you we'd talk about each of the different sets of secretion cells that are inside these gastric pits, and here we are. So here we have the mucosa cell here, the surface mucus cells. It's going to secrete mucus. This is really important because we want everything to continue to move along. We don't want anything to get stuck, so we want to make sure that things are churning well and they're mixing well. So these are the mucus neck cells here. They're also secreting mucus, but they're right at the edge of heading down, so they're no longer surface cells. They're at the neck cells. Underneath that, we have something that are called parietal cells. And parietal cells are very special. You know that they have this odd little organization where we have 2 different sections, and that's because we're secreting hydrochloric acid, and we're secreting it by secreting H on one side and Cl on the other in little pieces that are immediately going to become hydrochloric acid. And so this unique shape is going to be really specific for its function. It also is going to secrete something called intrinsic factor. Chief cells secrete pepsinogen and gastric lipase. Again, lipase is going to break down lipids. And G cells are going to secrete the hormone gastrin. And we'll talk about what each of these do in just a little bit. So this is that parietal cell that I told you had a really specialized form. And I'm not going to ask you to understand all of the different steps that are involved in these pathways. But what I want you to understand is that one side of the membrane, one side is going to secrete chloride ion, and the other one's going to secrete the hydrogen ion. Now, the chloride ion doesn't actually cost any money like ATP, cellular energy, but release of the hydrogen ion does. It costs 1 ATP molecule converted down into ADP, but what it does, and it also is going to bring in a potassium ion, but what it does is going to secrete that hydrogen. And again, that hydrogen and that chloride come together and make HCl. So this cell is a specialized cell that's constantly secreting HCl. All right, so what are the different types of cells? So first we have those surface mucus cells and the mucus neck cells. Both of them are responsible for secreting mucus. They're also involved in absorption. Parietal cells secrete intrinsic factor and hydrochloric acid. Hydrochloric acid is really important because getting that low pH is going to help kill any microbes that we might digest in our food. It's also going to start the process of chemical digestion by denaturing different proteins, and it activates a precursor called pepsinogen into that active pepsin, which is what's going to help break down proteins inside the stomach. We also have chief cells. Chief cells secrete pepsinogen and gastric lipase. As you may remember, lipase breaks down lipids. Pepsinogen is going to be that precursor to pepsin. So chief cells create pepsinogen, parietal cells convert them into pepsin, which is the activated form, which is responsible again for breaking proteins down into smaller peptides. G cells secrete gastrin, which stimulates the secretion of HCl by parietal cells, also stimulates pepsinogen secretion by chief cells, and contracts the lower esophageal sphincter. So it says no more food, right? Increases motility of the stomach and relaxes the pyloric sphincter to allow the food out into the small intestines. Muscularis layer is just responsible for peristaltic movement. It's going to be churning and physically breaking down the food, and then eventually help forcing that chyme through the pyloric sphincter, which is going to open and allow the chyme or chyme into the duodenum. Duodenum being the very first part of the small intestines. And the pyloric sphincter also helps prevent backflow of that chyme into the stomach from the duodenum. All right, let's talk about a very important accessory organ for a moment here. This is the pancreas. The pancreas lies just posterior to the stomach and produces specialized enzymes that help digest things like carbohydrates, proteins, fats, and nucleic acids. It also produces sodium bicarbonate. You may recognize sodium bicarbonate as a buffer that helps buffer your bloodstream. It's also going to help buffer your stomach acid. Remember, your stomach acid is coming out at a very low pH, and if we can help bring that pH back up to 7 very quickly, we can start the process of digestion in the small intestines faster. So the pancreas is going to store these different biomolecules until they're needed for digestion, so basically until the duodenum receives some chyme from the stomach, in which case it's going to empty its contents directly into the duodenum. So here's the pancreas itself. Here's the tail of the pancreas, the body of the pancreas, and then something called the pancreatic duct. The pancreatic duct is where we're going to be secreting all of our pancreatic enzymes. The pancreatic enzymes are going to, here's the head of the pancreas, going to secrete out of this region, so the very tip of the pancreas, into the duodenum by the major duodenal papilla, right next to the common bile duct. And we'll talk about the bile duct in just a minute. The bile duct connects up to the gallbladder, which then connects into the liver and is going to release bile. So both of these things are going to be released right into the very beginning of the duodenum to help do a couple of things. One, bring the pH back up to a reasonable level, and two, some digestive enzymes, add some digestive enzymes and different proteins to be able to help with the digestion in the small intestines. Okay, so this is an overview of what I just talked about. This here is the liver. The liver is connected by the falciform ligament. The liver is going to create the bile, which then gets secreted out by the hepatic ducts through the cystic duct into the gallbladder. The gallbladder is a storage organ. The gallbladder does not create bile, it simply stores it. And just like we saw previously, here's the fundus, again the fundus being the opposite region from the opening. This is the body, and then the neck of the gallbladder. And just like the pancreas, once it receives a signal from the small intestines that food has come out of the stomach in the form of chyme, then it's going to release that bile out here into the duodenum from a region that's going to connect. It's called the hepatopancreatic ampulla. So it's going to connect the hepatic region, right, which heads back up to the liver, to the pancreatic region, which heads to the pancreas. Again, here we have the tail, the body, and the head of the pancreas, and that's all going to secrete out the hepatopancreatic ampulla. However, we also have another accessory duct of the pancreas. So we have two different regions whereby the pancreas is going to empty into the duodenum. This is going to be the duct of Santorini. Right? This is an up-close view of the sphincter of the hepatopancreatic ampulla. It's called the sphincter of Oddi. Basically what happens is bile comes down through the common bile duct, and pancreatic enzymes come out through the pancreatic duct, and they meet here at the hepatopancreatic ampulla, or the ampulla of Vater, and then release into the duodenum through the major duodenal papilla. And this is what it looks like in an organism. Again, here's the liver, falciform ligament. This is the hepatic duct, which is going to be connected, of course, to the cystic duct, which connects up to the gallbladder. Those are all going to connect down into the major duodenal papilla, where we're going to have the common bile duct meeting with the pancreatic duct to be able to secrete both bile and pancreatic enzymes into the duodenum and the small intestines to be able to jumpstart the digestion in the intestines. So this is an overview of what happens with the liver and the gallbladder. So the liver makes the bile, and bile is really important in the emulsification of fats. So if you don't make enough bile, you may end up with too much fat coming through in your stool. So the gallbladder is going to store the bile until it's needed. So the liver makes it, the gallbladder holds on to it until the body says, hey, I need it. And this is kind of an overview of that. So here's the right hepatic duct, the left hepatic duct, and they come together to make the common hepatic duct, which comes from the liver. That's going to go back and forth to the gallbladder via the cystic duct, and then the common bile duct is going to meet up with the pancreatic duct to secrete into the duodenum, and then here's a sphincter, which is going to help maintain that inside until we need it. So the release is only going to occur when we have chyme entering into the duodenum. Okay, so the liver is composed of hepatocytes, canaliculi, and hepatic sinusoids. Okay, and that looks kind of like this. So we have a central vein where the blood's coming in, right? So the portal triad is going to comprise of the bile duct, the hepatic artery, and the hepatic portal vein, right? Inside here we have a bunch of hepatocytes, which form these hepatic laminae, and in between them we have the hepatic sinusoids. So the regions that are spaces are going to be sinusoids, and the regions that are going to have the cells are going to be the hepatic laminae. The cells themselves are called hepatocytes. We also have specialized cells here that are shown here in yellow. These are the Kupffer cells, or the stellate cells, and then that's all surrounded by connective tissue. So if you look at the liver histology, you can see here we have the central vein. Surrounding that we have the hepatocytes, and then we have the sinusoids, which are regions that don't have cells that are going to allow for blood flow. The portal triad, as I mentioned previously, that's the hepatic artery, the bile duct, and the hepatic portal vein, and they're kind of found in a triangle, and so they can have blood flow in, blood flow out, and then the bile all in one little section, so they're all constantly in communication with one another. And that's kind of what this looks like. So each of these is going to be, here's the central vein, here's the portal triad. This lobule is going to be between three of them, and then we also have an acinus in between the two of them. So there's three different units of liver structure and function. One is the hepatic lobule, one is the portal lobule, and then the hepatic acinus. Basically what happens is blood comes in from the hepatic artery, and then it's going to go into the hepatic sinusoids, go through the central vein, to the hepatic vein, into the inferior vena cava, and eventually back into the right atrium of the heart. right? We also are going to have nutrient-rich blood that comes in from the blood that's deoxygenated, coming from the hepatic portal vein. That's also going to go through that same thing for the hepatic sinusoid, central vein, hepatic vein, inferior vena cava, and right atrium of the heart. So we have blood that is oxygenated but does not have a ton of nutrients, and we have deoxygenated blood that is nutrient-rich, and they're entering into the same region that hepatic sinusoids, filtering through the central vein, hepatic vein, inferior vena cava, right atrium of heart. And that's important because although it was oxygenated when it came in, it was mixed with deoxygenated blood. So it's going to have to get right back into the heart to get oxygenated. And at the same time, what it's done is it's brought the blood that has the nutrients also back into circulation. Okay? So the function of the liver is several folds. One, it helps with metabolism of carbohydrates, lipids, and proteins. It also helps with the processing of things like drugs and hormones, the excretion of bilirubin, the synthesis of bile salts, which are very important in digestion. Also helps with storage. Again, this is the gallbladder we're talking about now. Phagocytosis and vitamin D activation also occur. So we activate vitamin D, and we can also attack any foreign invaders. So the liver and the gallbladder are actually very important in multiple different parts of your body systems, not just in digestion. All right, so we just talked about the small intestines. We talked about the duodenum anyway, in terms of things filtering from the stomach into the small intestines. So the chyme comes out of the stomach through the pyloric sphincter into the duodenum. From the duodenum, we're going to enter into the jejunum. From the jejunum into the ileum. So those are the three main regions of the small intestines. And then we're going to enter from the ileum into the large intestines. Again, ascending, transverse, and descending. Now, as we saw previously, like in the stomach, for example, we saw the rugged mucosa. We're going to see these different folds. These folds here are called plicae circularis. But basically the point here is that they're going to help increase absorption in the small intestines. So if food is able to pass over and around each of these, there's more surface area for absorption to occur. So what we previously saw as gastric pits are now evolving more into these villi. So we're still seeing the major layers, right? Mucosa, submucosa, muscularis. Again, we have lost that oblique layer of the muscle, so we only have two again. We have circular layer and the longitudinal layer of the muscle. Above that submucosal layer, this is going to be where we have very major role played by the lymphatic system, where it's going to be picking up all of the fats that are going to be pulled across. So these regions here are actually called a lacteal, and a lacteal is going to have a lymphatic capillary in the center. Remember, lymphatic capillaries are kind of one-ended. They're not kind of, they are one-ended. So it's not a connective system, or it's not a closed system. It's kind of a one-ended leaky system whereby things leak in through here and eventually end up in the lymphatic vessel. Surrounding that lymphatic vessel, we have a lot of blood supply. So we have oxygen-rich blood that's going to also be delivering that blood to these tissues, and then oxygen-deprived blood that's going to be picking up a lot of the different nutrients. So it's going to be oxygen-deprived, but nutrient-rich. We have a couple specialized cells here, similar to what we saw in the stomach. These here are goblet cells. We also have an enteroendocrine cell here, and all of that is going to be surrounding, again, this lamina propria region, which is where the lacteal is going to be found. And the entire thing is called a villi. Right? So we have these specialized villi at the top of the villi, we have microvilli, and all of that is going to be involved in digestion and absorption. So instead of having these gastric pits, we now have a much more open region. We have these intestinal glands that are going to secrete different intestinal juices, and we're going to allow all of the food material to go through all of these villi. So instead of having pits where we're secreting things out, now we're allowing the food material to pass through. We have intestinal juices that are going to be secreted by these intestinal pits here, these intestinal glands. This intestinal juice is going to help provide a vehicle for absorption of multiple different substances from the chyme. Things are going to get picked up from the chyme as they come in contact with the villi. The villi have specialized regions called microvilli on the end, and those contain something called brush border enzymes. Brush border enzymes are specifically made to help break down food products to allow for increased absorptive rate. So this is kind of like what that looks like. Again, we have the muscularis, the submucosa, the mucosal layer. In this case, the mucosal layer is broken down up into these villi regions. Right, these villi are going to be on top of intestinal glands that are secreting intestinal juices. And if you look at the tips of these villi, they actually have microvilli on top, which aren't really shown here. And they're going to have brush border enzymes on the tip of those that are going to help digest the food. Digested food products are going to be picked up by these absorptive cells. We also have the specialized goblet cells, which are secreting cells. And then again, this is the lamina propria, which is found inside here. These are the intestinal glands, which are going to secrete the intestinal juices. We have two different forms of mechanical digestion that are occurring in the small intestine. So most of what's happening is chemical digestion, but we do have mechanical digestion, including peristalsis, which is contractions that help propel things forward, and then segmentation, which are contractions that help kind of churn things up but stay in place. In the small intestines, we're going to continue with the digestion of carbohydrates. Remember, that started in the mouth. We're also continuing with the digestion of proteins, and that started in the stomach. Continue with the digestion of lipids, that also started in the mouth, and the digestion of nucleic acids, which started in the stomach. So the chemical digestion of the small intestines is really just continuing what got jump-started earlier on in this pathway. So the organs that are the accessory organs include the pancreas, the liver, and the gallbladder, right? The pancreas is responsible for delivering pancreatic juice into the duodenum. It goes through the pancreatic duct, and it goes right at the same region where the bile from the liver and the gallbladder are going to go in. The liver creates bile or bile salts that helps break down and emulsify and then absorb the lipids. Gallbladder is going to store that bile, concentrate that bile, and then secrete that bile through the common bile duct to the duodenum when necessary. Small intestine is going to be the major site of digestion and absorption. We're going to be responsible for secreting intestinal juice that comes from the intestinal glands. The absorptive cells are going to digest and absorb nutrients. Goblet cells secrete mucus. Enteroendocrine cells are going to secrete specialized hormones like secretin, cholecystokinin, glucose-dependent insulinotropic peptides. We'll talk about them a little bit later on. I'm not going to go too heavily into depth with those, though. We also have Paneth cells, which secrete an enzyme that's bactericidal called lysozyme, which is going to help with your immune response, and also help with phagocytosis. So anything that it does kill, it's going to want to digest. We also have duodenal glands or Brunner's glands that help secrete alkaline fluid. Remember, it came out of that stomach really acidic, so we're going to buffer that stomach acid. Also create some mucus to help lubricate and protect that intestinal layer from anything that might be running across it, particularly in that region where we just dumped a bunch of acidic juices from the stomach. Remember, we have specialized circular folds. The more folds you have, the more surface area, more digestion, more absorption. We have villi. At the tip of the villi, we have microvilli, so we have finger-like projections that have tiny little membrane finger projections at the tip of them that contain something called brush border enzymes. Brush border enzymes are going to help break down food products, then also help increase surface area for digestion and absorption by having more surface area on the microvilli. And the different types of muscularis, we have the segmentation, which is one of the types of peristalsis, which we have two different types of contractions, right? We can contract forward or we can contract in place, and we alternate back and forth between these to allow both mixing and propulsion. And then we have that migrating motility complex. That's that propulsion that I talked about. That's a specific type of peristalsis where we have a wave of contraction. So A squeezes, then B squeezes, then C squeezes, in which way all of the food matter and waste is going to be moved forward. So it's a series of contractions and relaxations of the muscle inside the intestines that allow the chyme to move forward toward the ileocecal sphincter. That's called the migrating motility complex. Some of the enzymes that we have found or discussed or we've talked about through this project is the saliva. In the saliva we have salivary amylase found in the salivary glands, and it breaks down starches into disaccharides. So poly into disaccharides. Again, later on we're going to start talking about dyes into mono, right? So disaccharide to monosaccharide, or dipeptide to a monopeptide or something like that. So we also have lingual lipase, which is going to also come from the saliva. It's going to, as you might imagine, it's a lipase. It's going to break down fats, so it's going to break down triglycerides into fatty acids and diglycerides. So again, we're going from large to medium here in the saliva. We're not going from medium to small yet. Later on in the stomach and in the small intestines, we're going to be going from medium to small and then smaller yet. In the stomach, we secrete pepsin and gastric lipase. Pepsin, remember, is a specialized enzyme that breaks down proteins at a very low pH. So it only works in the stomach. When it moves into the higher pH of the intestines, it no longer works. Gastric lipase is going to break down triglycerides into fatty acids and monoglycerides. In the pancreas, we're secreting a ton of different digestive enzymes. The first is pancreatic amylase. Again, you recognize amylase as breaking down starches. We saw that previously in the saliva. That's going to be secreted by the acinar cells. Actually, they all are, so I'll save us some time. Trypsin, again from the acinar cells, breaks down proteins. It does the same thing as pepsin, but it does it at a different pH. So it does it in the intestinal pH. So it breaks down proteins into peptides. It breaks them down in a different region than pepsin does, but that's a kind of a detail. Chymotrypsin is going to be activated from a precursor molecule called chymotrypsinogen. It's activated by trypsin, so trypsin not only breaks down proteins, but it activates chymotrypsin if it needs assistance, basically, to help break down proteins into peptides. Elastase, same thing, activated by a precursor molecule called proelastase and breaks down proteins into peptides, also activated by trypsin. Carboxypeptidase is activated from a precursor molecule called procarboxypeptidase, again by trypsin. So trypsin is going to not only break down proteins itself, but activate chymotrypsin, elastase, and carboxypeptidase, which are going to break down proteins, and in this case, it's going to be breaking down peptides into amino acids and peptides. So carboxypeptidase actually breaks smaller into smallest. Pancreatic lipase is going to break down triglycerides into fatty acids and monoglycerides. And then we have some nucleases, which do exactly what you might expect. They break down nucleotides. So they break down ribonuclease. It's going to break down ribonucleic acid and nucleotides. Deoxyribonuclease is going to break down deoxyribonucleic acid. And you should recognize those as RNA and DNA, respectively. All right, some other enzymes that we've encountered include some brush border enzymes. These are found at the microvilli plasma membrane on the small intestines. Things called alpha-dextrinase, maltase, sucrase, lactase, enterokinase. All of these are breaking down, well, not enterokinase. Up until here, we're breaking down sugars. So we're breaking down alpha-dextrin into glucose, maltose into glucose, sucrose into glucose and fructose, lactose into glucose and galactose. So each of these are going to be breaking down a larger sugar into smaller sugars. And then here, these enterokinase is going to be breaking down trypsinogen into trypsin, so it's basically going to be activating trypsin, and as you might recall, once trypsin is activated, it's also going to activate chymotrypsin, elastase, and carboxypeptidase. We have some peptidases here, aminopeptidase and dipeptidase. These are going to be breaking down the amino acids off at the end of peptides, so just one at a time, or this is going to be breaking dipeptides into amino acids. Either way, we're getting from the point where we're now going from smaller to smallest. And then the nucleosidases and phosphatases are going to be breaking down nucleotides into their nitrogenous bases, pentose and phosphases. So pentose being a sugar and phosphates. These are going to be what is the building blocks of a nucleotide, right? A sugar, a phosphate, and the nitrogenous base. So how are these all absorbed? So they can be absorbed by multiple different ways. So let's just look at this pathway here first. They can be absorbed, or this column here. They can be absorbed by secondary active transports. They can be absorbed by facilitated diffusion. They can be absorbed by active transports, secondary active transport with hydrogen instead of sodium, simple diffusion for things like small fatty acids that are tiny enough to be able to go right across. Also, some of the larger ones that are able to be carried across in micelle form can go by simple diffusion. So there's a lot on here, and I'm going to walk you through each of it step by step. So let's start at the top. Glucose and galactose are brought in with sodium. So it's going to be active transport. Active transport means that we're going to be spending cellular energy. And that's basically because these guys are too large to make it across on their own, so they need to have specialized transporter molecules that are going to help get them across the border. Fructose is going to be kind of the same way, except it doesn't need to be active transport. It's going to go by facilitated diffusion, so it's not going to cost cellular energy, but either way, it's going to get across and it's going to end up in the blood capillary of a villus. And then these are going to end up being brought into the hepatic portal vein and then into the liver. These guys are all going to be taking a different pathway than this pathway here, so I'm going to talk about that pathway there last. But these three on the top, well, these 5 here on the top, these are all going to go in through the hepatic portal vein and then into the liver. Amino acids can be brought in by secondary active transport with sodium or by active transport. They're going to go right across. Dipeptides are usually brought in by diffusion or by secondary active transport. And then simple diffusion is what's basically going to be bringing in our short chain fatty acids. They're small enough to make it through, and then they're going to make it through across the other membrane by diffusion. But these fatty acids here and monoglycerides are hydrophobic, and that's a problem because the blood supply is hydrophilic. And so to be able to get it into the blood means it has to have a carrier molecule or form something that's called a micelle, which is going to have some amphipathic carrier molecules available to it. And so in order to do that, it's going to go in by simple diffusion across these brush border enzyme, microvilli, brush border, but it has to form this called a chylomicron. I know it looks kind of like a stoplight, but basically we have triglycerides that are going to form with the monoglyceride, or a monoglyceride forms into a triglyceride and then into a chylomicron, which then gets moved over to the basolateral surface into the lacteal. Remember I told you the lymphatic system was responsible for transporting all of your fatty acids. Then that's going to end up going through the thoracic ducts and then heading into the left internal jugular and left subclavian vein junction. So that's where it's going to end up returning back into the blood supply. So, I love that this says greatly enlarged, because this is microscopic and this is the human body. So it's obviously very greatly enlarged. This here is going to be the chylomicron that we talked about. So this is the way by which the triglycerides are going to be carried into through the lymph into circulation. All right, so basically it's going to go across the villus into the lacteal, into the lymphatic system in the lacteal, and then get transported out, I apologize, through the lymphatic system, through the thoracic duct, eventually meeting up into the left subclavian vein, and then heading in that way into the circulation. Now, some of the other molecules that are water soluble, including these short chain fatty acids, amino acids, monosaccharides, etc., they are going to be picked up by the blood supply. So remember, I previously said that lacteal region has highly oxygenated blood coming in, and then deoxygenated blood that has a lot of nutrients in it coming out. So no nutrients coming in, but oxygen. That oxygen gets delivered to these tissues, it picks up the nutrients, and then heads back out. Now it's going to go into the liver. It's going to go into the liver through the hepatic portal vein, and then go through that hepatic triad that we talked about, and then eventually end up back into circulation through the liver. Let's talk about fluid balance for a second. So over our entire day, we are creating and then reabsorbing a little over 9 liters. So we are ingesting some of that, we are creating some of that. So saliva, we're making approximately 1 liter a day that gets swallowed into our stomach. We're making approximately 2 liters of gastric juice a day. We're supposed to be drinking approximately 2 to 3 liters of liquids a day. Additionally, we're making 1 liter of bile, we're making 2 liters of pancreatic juices, and 1 liter of intestinal juices. So the total that we are ingesting and secreting is approximately 9.3 liters in and approximately 9.2 liters out. And only 0.1 liter is actually going to be excreted into feces. Why? Because the role of the small and the large intestines is water resorption, right? We want to make sure that we are compacting that fecal matter down so that we give it the minimum amount of water necessary to be able to be smoothly excreted. The rest of it needs to be resorbed back into our body. All right, we talked previously about the villus inside the ileum, so the ileum means the small intestines. So the villi here are going to be facing the lumen of the ileum. Underneath that, we have these lymphatic nodules. So lymphatic nodules are going to be responsible for eliminating any foreign invaders. And the intestines is going to be really susceptible to foreign invaders. Whatever has survived the stomach is going to head directly into the intestines. So in that submucosal layer, we're going to have these large lymphatic nodule regions, which are going to be responsible both for the inflammatory response and also for the acquisition of these different chylomicrons that are then going to have to end up back into circulation. All right, after we travel through the small intestines, then we enter into the large intestines. So this is the overview of the large intestines, it's also known as the colon. Here's the ileum. This is the end of the small intestine. So the ileocecal canal is going to lead into the cecum through the ileocecal sphincter, or different valve. And now we're in the cecum, we're going to head up into the ascending colon, the transverse colon, the descending colon, the sigmoid colon, rectum, and the anus. You'll also see these little sections here that look like little circular sections. Those are called haustra. And you will also see a lot of little yellow, these are attachment points for the omentum. So these are the omental appendices. So this is going to be how these different regions of the organ are going to be held in place in the abdominal cavity. All right, let's see. So if we're actually looking inside the anal canal, here we have the rectum and then the anal canal. We have 2 different sphincters. You may remember from the urinary system that we had 2 different sphincters there too. We have an internal anal sphincter and an external anal sphincter. One of these is involuntary and one of these is voluntary. So that internal sphincter is involuntary. So when waste products move from the rectum through the anal canal and get pushed into this region here, that's an involuntary response. And that's when you get the urge to use the bathroom, but you still, thanks to this external anal sphincter, have time to make it there on your own accord. All right, so the functions of the large intestines are going to be haustral churning. You saw those little regions here were called haustra. Basically, they're going to be churning. Each one of those are going to have sections inside of them where they are churning around. We also have peristalsis and mass peristalsis, which are moving things forward. So we're moving both in place. Picture this like stirring a pot on the stove, and moving things forward. Picture this like pouring cement out of a cement truck. And that's going to help drive contents forward from the colon into the rectum. We also have specialized bacteria here. Bacteria in the large intestines are going to be converting proteins into amino acids. So there's going to be certain things that our body can't do ourselves, but we use bacteria in a symbiotic way to be able to convert different proteins to amino acids that we can't do on our own, break down amino acids that we're unable to digest on our own, and then also production of certain vitamins like B vitamins and vitamin K that we're unable to provide in our body. Last but not least, the absorption of water, ions, and vitamins. Remember, we want to remove as much water as possible to be able to form feces that's going to be able to defecate smoothly, but not going to be too liquidy because we want to retain that liquid inside our body. And then the actual act of removing that feces from our body through the rectum is called defecation. All right, so here's the histology of the colon. So just like previously, we have the mucosa, the submucosa, the muscularis. But in this case now, we have little openings of intestinal glands, and we're basically just responsible for absorption. So here's we have absorptive cells, we have goblet cells. Remember, these secrete mucus. Intestinal glands are going to secrete that mucus. We're still seeing the lamina propria, which is surrounding the lymphatic tissue, and then also, again, the arteries and veins. The veins are going to be picking up all of the, it's going to be deoxygenated, but picking up a lot of nutrients. Arteries are going to be supplying oxygen. These guys here, lymphatic nodules, that's that mucosa-associated lymphatic tissue that I talked about. It's going to be responsible for our immune response. So again, the lymphatic system not only does the immune response, but also picks up fatty acids. All right, inside these glands, we have absorptive cells, which are responsible for absorbing water. They have these microvilli regions, which are very good at drawing water across. We also have goblet cells that secrete the mucus. So we're basically secreting mucus and absorbing water as we move along. And that looks kind of like this underneath the light microscope. Here's our mucosal region, our submucosal region, muscularis and serosa. This is going to be the lumen of the intestines. Here we have the lamina propria, which is found inside what used to be microvilli when we were, or used to be villi when we were in the small intestines. But now, actually, that's more solid, and we're just going to have these little holes at the top. So the fecal matter doesn't go in anymore. Instead, things are secreting out, and it's running along the top. This is the opening of the intestinal gland. The intestinal gland is going to be comprised of goblet cells, absorptive cells, so it's going to be secreting mucus and picking up water as we go along. And this is that lamina propria, which is going to be the region in between, where we're going to be absorbing any nutrients that are coming across and putting them back into the blood supply or into the lymphatic tissue. So we talked a lot about enzymatic digestion, but I haven't talked a whole lot about the mechanical digestion. I kind of touched on it. There's a couple different sets of mechanical digestion that's occurring in the colon. First, we have that haustral churning. Remember those regions, the little polyps are called haustra. And what happens with the haustra is that they distend. So they get larger and larger and larger, and then they can't handle it anymore, and so they squeeze. And when they squeeze, they both mix everything around internally, like stirring a pot, but they also force things forward a little bit as well. And that creates peristalsis, or propulsive contractions, which are going to basically mean A squeezes to B squeezes to C squeezes to D, etc. And we also have something called mass peristalsis, where it all works in concert. That's a quick wave beginning in the transverse colon and quickly drives the contents of the colon into the rectum. And we've all felt this. This is going to give us the spontaneous urge to defecate. Basically, that internal sphincter is going to give way, giving pressure to that external sphincter, which is going to mean that we're going to have to find a bathroom post-haste. The very final stages of digestion are going to occur by the use of bacteria. So we have good bacteria in our gut and bad bacteria in our gut. In fact, we have determined now that a lot of different intestinal disorders like Crohn's disease, etc., are linked to gut flora, and some extreme measures include things like fecal transplants, where they'll take fecal matter from a healthy individual and pop it into an individual who's suffering from intestinal distress, and hopefully be able to regain normal bacterial flora. So the bacteria do a lot of things. They break down several different substances, but they also synthesize things, so they make vitamins for us. Another things that happen in the main thing that happens in the colon is the absorption of water, electrolytes, and then whatever residual vitamins are there, and the creation of feces, which consists mainly of the inorganic salts and bacteria, but also some soft epithelial cells as we've passed through the GI tract, leftover bacteria, and residual undigested portions of food. Last but not least, we have this defecation reflex, whereby the rectal wall is going to descend. We have stretch receptors inside there that send information through the spinal cord, so these nerve impulses that then result in motor impulses, and motor impulses travel back through the descending colon, through the sigmoid colon, the rectum, and the anus, so it travels to the entire lower digestive tract. And then we end up with the contraction of the longitudinal rectal muscles, and that's going to open the internal anal sphincter. And when that internal anal sphincter opens, everything is going to get dumped into the rectum and put pressure on that external anal sphincter, which needs to be voluntarily relaxed, allowing for defecation to occur and the expelling of the fecal matter. So just to summarize what happens in the large intestines. So inside the lumen we have bacteria that are going to be found here. The bacteria are going to assist us in digestion by breaking down undigested carbs, proteins, amino acids into different products that can either be absorbed and detoxed by the liver or expelled in the feces. It also synthesizes specialized vitamins, right? B vitamins and vitamin K. So certain things that we can't create ourselves and would have to take exogenously otherwise. The mucosa inside the large intestines is responsible for secreting mucus, so those goblet cells, and then absorption of water, which is going to help solidify our feces and help retain the body's water balance, and also absorption of solutes that we can acquire here, including ions, and then whatever vitamins are going to be created by the bacteria that we want to resorb. And then the muscle layers do a couple of things, right? They churn things around. So we have haustral churning inside each of the haustra, which moves contents from one to the next through muscular contractions. We have peristalsis, which move things along the entire length by coordinated contractions of both circular and longitudinal muscles. And then that mass peristalsis, whereby everything moves all at once, forcing the contents into the sigmoid colon and into the rectum, and therefore putting pressure on that external anal sphincter, which leads to that defecation reflex, whereby we eliminate the feces by contraction on the sigmoid colon and the rectum. So this is just a summary of all of the different organs of the digestive system. So the tongue is responsible for mastication and making the bolus of food swallowed, so deglutition, so basically chewing and swallowing. Also for taste determination and the initial digestion of things like triglycerides. So it's going to help start to break things down. We also have salivary glands that produce saliva. Saliva is responsible for moistening the food, softening the food, dissolving the food. Also helps clean your teeth. It's going to have some antimicrobial agents. And initiate the digestion of starch through amylase, which breaks down amylose, which is the sugar. ACE is the enzyme, OS is the sugar. The teeth are responsible for mechanical digestion, cutting, tearing, pulverizing food. The pancreas is responsible for creating pancreatic juice. That's going to do several things, including buffering the acidic gastric juice in the chyme, stopping pepsin from working, adding in trypsin or creating trypsin, creating the proper pH for digestion, and then also the digestion of carbohydrates, proteins, triglycerides, and nucleic acids. So pancreatic juices are responsible for a lot of assist in digestion. The liver creates bile, which is then stored and concentrated in the gallbladder. What does it do? It breaks down, so emulsifies and absorbs lipids through the small intestines. The mouth is going to be responsible for it's basically the first place that all the food goes, and to help keep food inside the teeth through mastication and assist with chewing and swallowing. Pharynx is the first area that the food goes into now that it's a bolus from the oral cavity as it passes into the esophagus. Through the esophagus, it's then going to go down into the stomach. The first thing it's going to have to hit is that upper esophageal sphincter that's going to be at the top of the throat, and then eventually it's going to have to hit the lower esophageal sphincter, which is the door that's going to allow the food into the stomach. Inside the stomach, we have these waves of contractions that are going to mix the food with the gastric juice and saliva. We're going to activate pepsin, start digestion of proteins, hopefully kill off all of the microbes in the food, and start absorbing some vitamins, so vitamin B12, etc. That also is going to contract off the lower esophageal sphincter. Sorry, I couldn't take another bite, so no more food can come in. Allows the stomach to have a lot of motility. Oh, and not just the no more food can come in, right? We can end up with a series of gastric juices that can come up into the esophagus with gastric reflux, what can end up damaging the lower esophagus and be very painful. So we want to make sure that we're keeping the gastric juices separate from the esophagus. Last but not least, it's going to relax that pyloric sphincter when it's ready, and that's going to help release chyme one little ball at a time into the small intestines. Small intestine is going to start mixing the chyme with the digestive juices. We're also going to get our pH back up, so we're going to now be using a different enzyme to break down our proteins. Going to move the chyme towards the ileocecal sphincter in different regions, so different sections, moving one step at a time until we get to the ileocecal canal. And also we have a lot of digestive secretions in there that come from the small intestines, the pancreas, the liver, etc. So we're now going to be continuing the digestion, actually basically completing most of the digestion, things like carbohydrates, lipids, proteins, and nucleic acids. Large intestines is going to be responsible for water resorption, and then also the last sets of haustral churning, peristalsis, absorption of water, vitamin K, production of the B vitamins, and then also the resulting resorption of those B vitamins, and then last but not least, defecation. All right, so how is digestion regulated? So we have three major regions. We have the cephalic phase, which is basically cephalic being head or brain, so it's going to be neuronal and helps stimulate gastric secretion and mobility. We also have the gastric phase, which uses both neural and hormonal mechanisms, and the intestinal phase, which is regulated by neural and hormonal mechanisms as well. Okay, so this here is a feedback loop, and just like any feedback loop, we're going to have homeostasis that occurs, then is going to be disrupted. In this case, homeostasis is disrupted by the food entering the stomach. So the controlled condition is the pH of the gastric juice, which is going to change. You got to figure the pH of the gastric juice is low to begin with. When food enters it, it's going to increase a little bit. Also, we're going to have stretch receptors, noting that we have a distension of the stomach walls. Both of those, right, these receptors that are the chemoreceptors and the stretch receptors, are going to send information to the control center. In this case, the control center is in the submucosal plexus. So that's going to have nerve impulses. Remember, these are the parasympathetic nerve impulses that are occurring. If you figure that fight or flight is sympathetic, rest or digest is parasympathetic, we're completely on the rest or digest side here. So these are parasympathetic impulses that are going to regulate that submucosal plexus and affect on these specific cells, namely the parietal cells, which are going to create HCl. So we're going to be secreting more of the gastric HCl. We're also going to be contracting our muscles in the stomach, and this is shown from multiple directions because remember we have that third muscle layer there, so it can contract in 3 different directions. So the response is that we're going to increase the acidity because we've secreted the HCl. We're going to start mixing the stomach contents and eventually start emptying that contents out into the small intestines. And when we finally return to homeostasis, because the distension is back to normal and the pH is back to normal, basically pre-eating status, we've made all the food out of the stomach into the intestines. Now our controlled condition is back to normal and we have stopped our negative feedback loop. So these are some of the hormones that we spoke about previously that are responsible for controlling digestion. The first is gastrin. Gastrin is stimulated by stomach distension and by the receipt of partially digested proteins, also by caffeine. So you can stimulate gastrin just by having a cup of coffee. Also by high pH of stomach chyme. That's all of that's going to stimulate the gastric secretion, and that comes from the enteroendocrine G cells, which are found in the pyloric antrum of the stomach. What are the effects of this? It's going to secrete more gastric juice, increase the gastric mobility, so your stomach's going to move more, and promote growth of the gastric mucosa. This is important because the gastric mucosa starts to get broken down by the hydrochloric acid, so we have to make sure that we replace it quickly. Some of the other effects is that it's going to constrict that lower esophageal sphincter. Sorry, I'm so full I can't take any more food in, and relax that pyloric sphincter to allow food out into the small intestines. Another hormone we'll talk about is secretin, which is stimulated by acidic chyme entering the small intestines. So when the small intestines first receive the food from the stomach, it's very acidic. And so that's going to release or induce the secretion of secretin. And that's also going to happen by enteroendocrine cells. But in this case, it's going to be in the mucosa of the duodenum, so not in the stomach itself. The effects of secretin is that it stimulates secretion of pancreatic juice and bile. So all of a sudden we're going to have a ton of bicarbonate atoms. Remember, bicarbonate ions are going to help increase the pH, so bring it back to a more normal level. Also going to help inhibit the secretion of gastric juices. It's telling us we no longer need it, right? The food's coming from the stomach into the small intestine, so we probably don't need any more gastric juice. And also it's going to help maintain the pancreas and enhances the effect of CCK. CCK is cholecytokinin. And that's actually going to be stimulated by digested proteins, partially digested proteins and amino acids and fatty acids that come into the small intestines. And that's going to be stimulated or released by these enteroendocrine cells that are going to come from the mucosa, in this case of the small intestines. It's also released in the brain. So this is something that can have some salivary effect, kind of like a Pavlov dog kind of effect. So CCK is going to stimulate the secretion of pancreatic juice, rich in these digestive enzymes, also causing ejection of bile from gallbladder and opening of the sphincter into the hepatopancreatic ampulla. Remember, that's going to be the region that's going to dump right into the duodenum. It's also going to induce satiety, which basically is the feeling of satisfaction. I'm full. I don't want another bite. No, thank you. I know that looks delicious, but I couldn't possibly have another. One of the other effects is the inhibition of your gastric emptying, so it's going to slow down the stomach emptying. Also promoting normal growth and maintenance of the pancreas and enhancing the effects of secretin. So you can see these guys are going to work hand in hand. This one enhances the effect of CCK. This one enhances the effect of secretin. All right, I'm going to gloss over this because we did give you an entire development lecture, but the digestive system is actually going to be formed earlier on in digestion, about the earlier on in development, around the 24-day mark. The primitive gut is formed from the yolk sac by a process called gastrulation, which is basically making this midgut section. And you can see this whole section here is basically going to become the digestive tract. So here we have a transverse section, these are the sagittal sections, and it's starting to become a long tube here by the 28-day mark. Here's the oropharyngeal membrane, which is going to be the oral pharynx, right? So the head, cloacal membrane, okay, tail. This is basically going to be becoming the mouth and eventually becoming the anus. All right, as we get older, aging can result in many effects on the digestive system. The first, that we have decreased secretory mechanisms and motility. So we're not going to be able to secrete as many of the digestive juices. We're also going to have loss of tone of the digestive muscular tissue. So just like any muscle starts to atrophy and wear down as we age. And we won't be as responsive to our neurosecretory feedback as well. So some of our homeostatic conditions that are going to bring things back to normal might not be as responsive. Also don't have the same response to internal sensations and pain. So this is basically just showing you how the digestive system contributes to all of the different body systems. And basically the point of the digestive system is to break down the dietary nutrients that are going to come in through the food in ways that we can absorb them and use them to build ATP and other building blocks for the tissues, as well as absorbing back minerals, water, and vitamins needed for growth and function, and elimination of waste from the body, tissues, and feces. So thank you guys for sticking with me this long. I really appreciate it. I hope you guys have a fantastic day.